The biological clock is a fundamental adaptation animals, plants, and fungi have. It allows them to adjust to the daily light-dark cycle of the planet. We focus on a very simple organism, a model system in genetics called Neurospora crassa. And in this organism, the biological clock controls its reproduction. And what we see here, as the organism grows down the tube, these little bands, and that's the organism reproducing. And that reproduction takes place every 24 hours. Most experiments that are done on the biological clock have involved measurements on millions and millions of cells. Various strains of this organism have been engineered so that we can actually look at the behavior of the clock in individual cells. The challenge, though, of doing this is those cells are floating around in a solution, and so it makes it very hard to track what they're actually doing.
So what we're doing is we're using something called microfabrication. The same process people are using to produce CPUs, to produce integrated circuits. Instead of producing the electronic device, we're using that process to produce a fluidic device. We call this a microfluidic device. And with that kind of device, we can basically generate many different droplets, and we can use these smaller droplets to trap individual cells inside them and put them into a long-term observation device and observe their behavior over a very long period.
The solution of this problem of reconstructing the biochemical and gene regulatory networks in the cell requires a multidisciplinary approach, because we're using computational algorithms and techniques from statistical physics, from statistical mechanics, genetic methods to identify and track the expression of genes in the cell. It requires an engineering approach to build these microfluidic devices. And lastly, it also requires input from computer science, because the simulations we do actually are so computing intensive that a very high level of computational parallelization is necessary.
In this research, we'll be able to predict function of genes that consist of 2,436 genes within a few days instead of more than 12 months.
Previously, we used just sequential algorithms, but now we were able to use this parallelization algorithm using GPUs. For some algorithm, this is actually very fast compared to the sequential algorithm. We might be able to get, depends on how many GPUs you're going to use, 400 times speed up or even more.
In our research, two stages of the GPUs. The first stage we used GTX 480, which consists of 480 cores, and in the later stage, or in the production stage, we used Kepler 20X GPUs, which is the newest GPUs in the world, and these GPUs consist of thousands of cores in it.
So aside from the scientific impact of this work, namely that it will allow us to get a deeper and more profound understanding of how the cell produces its behavior that we observe at the molecular level, this work will of course also impact things like drug design, because you understand the mechanisms of the microbial organisms better by using these methodologies, and therefore can design drugs that interfere with it. And also it will have impact on human biology and perhaps ultimately medicine.